this is glycolysis. Glyco means sugars or sweet, and lysis means breaking down. So glycolysis is literally physically the breaking down of sweets. Glycolysis, say it with me, is a anaerobic pathway or an anaerobic type of work, right? Which means it does not require the presence of oxygen. It's anaerobic. Okay? Let's see how glucose is broken down. So, glucose, we have 6 carbon figure called glucose. And this all happens in the cytoplasm. It happens where? In the cytoplasm of a cell. Where? Cytoplasm. Okay. So we have a 6 carbon compound, which is glucose. And then in the cytoplasm, our cells have to spend, just like an investment, our cells have to put a little bit of energy in the form of 2 ATP in order to drive this process, in order to start the process, in order to make the ATP needed for our bodies to survive. So this 2 ATP is going to get converted into 2 ADP. And those 2 phosphate groups, because you break it down into ADP and 2 phosphate groups, those get added onto the glucose. This over here, this structure is a very unstable structure. So this unstable structure is going to break right in the middle and make 2 G3Ps, okay? These are, and it's 2 different pathways, but necessarily the same thing happens, right? So we have a 3 carbon compound and then a phosphate group. During glycolysis, 1 molecule of glucose, which is a 6 carbon compound, is transformed into 2 molecules of pyruvic acid, which we're gonna see here later on. So how does it work? So now that we have 2 of the same G3Ps, inside the cytoplasm, our cytoplasms are not just empty space. They're made of organic compounds, hydrogen, like ions, and polar molecules, things like that. So we have phosphate groups found in our cytoplasm. So 1 phosphate group is going to get added on to the already G3P, and then you're going to get this structure where it's a 3 carbon compound with 2 phosphate groups. And then in a series of reactants, NAD plus, which is already found in our cytoplasm, combines and reacts with 2 electrons and hydrogen ions found also in the cytoplasm, and they make NADH. So it removes, like one of the reactants of glycolysis, like this, shown by this arrow, is showing basically how they're removing electrons, which are high energy, and then they are passing them to NAD plus, and then also hydrogen ions, and then or protons, and then they're making NADH. And then this is held on and then sent to the electron transport chain, which I'll make another video later. And then another series of reactants is where we have 2 ADPs, and then it's going to be added plus 2 phosphate groups, and then we're going to make 2 ATP. So on one side, there's 2 ATP being made and 1 NADH being made. And then after these reactants happen, what is left after all the reactants is a 3 carbon compound named pyruvate. So basically, the cell, as we saw in the beginning, the cell will invest or put in energy to get more energy later on, because we put in 2 ATP, but in total we made 4 ATP, because we see 2 here and 2 here, right? And two reasons to do this, and two advantages of glycolysis is to trap glucose, and it's like easier to break down. And NAD plus right here that we see, it has the ability to strip the electrons from this, or the G3Ps, and hydrogen ions, and it makes NADH, which I already said that. So this occurs in both prokaryotic and eukaryotic cells because it does not require organelles or oxygen, because it happens in the cytoplasm. That's why, because prokaryotic cells also do have cytoplasms, so prokaryotic cells will also see glycolysis, but then after they will go through a different process. That's basically it for glycolysis. After glycolysis has happened, 90% of the energy stored in the original glucose is still there. Only 10% of it was released in the form of like the NADHs and the ATPs, right? And it's still not glucose. Now this glucose is transferred into 2-pyruvates. And this will go through the precursor steps and then Krebs cycle and then the ETC. So yeah, that's it.